pretty good. I haven't been doing my homework, so
You've been behind a little?
yeah, well, a lot, not a little.
That's okay. I'm starting the recording, okay? So don't worry.
Okay.
But don't, Grisha, where are you going? But don't worry, because a lot of people are also behind. And it's time, it's sort of the last stretch of the course that we want to catch up. And if you'd like to get some support in the planning or so, then reach out, let me guide you a little bit, and we can have a planning session or so, so we can get this successfully finished up and taken care of in terms of the class goals. Are you working on the pathology project with your group?
Yes, I'm with Catherine and
Tranisha?
yes, that's right.
2 Hour Livers is your group name. That's a fantastic one. So that's moving along. You guys are connecting with each other?
Yes, we have been connecting, and so we're gonna have a like we're gonna do our, so there are 3 questions. We're gonna do each question, and then we're gonna put all together into a PowerPoint presentation. Yeah,
correct. And so you're on, that's good. So you guys are on target with that. And then when you guys done some research and a little bit ahead, then you started doing your PowerPoints, then let's have a meeting together, and I have a look at it as a sort of a draft in a group. We do a group Zoom together, and then we can work on making a second draft, and then once we're done that, we can make a presentation. Does that sound all right?
Yeah, so am I right that you want to see our draft, PowerPoint draft sometime? When would be like before we submit it this Sunday? Is that right?
Yeah, just start when you have something, start uploading a draft PowerPoint, so I can start looking at it. If you want to just send me a link with a Google whatever text, send me over text, that's fine. If you feel like you have some and you'd like some feedback, that's when you got to involve me, and we have some. At least I want to have one session with each group to just sort of go back and forth a little bit. Usually it ends up being the wording a little bit, and maybe the depth of the research, and then most likely it's to add pictures to the PowerPoints, because at the end I want you guys to make a little video and explain something. So it's always good to have pictures to explain.
Okay, so so this is not like we're not gonna present it in class, but we're gonna make a video, like a voice over PowerPoint. Is that right?
Yes.
Oh, okay, got it.
Yeah, you kind of like I do in Zoom. You go on Zoom, you take my ID, or you take I have one for the class. You go on there, everybody, and then you press record and you share your screen, and then the person, you go one by one, and that way you make a little, let's say, I think 6 to 8 minute clip of explaining a pathology, and then other students can, your peers can read it and learn something from it, and they learn something about the pathology, and then they can give you some feedback. How was that for them to learn that from you? And you can then have a few pointers for next time, improve on that, because it's it's always a process.
Yeah.
And so that way we do a few things at once. That makes some sense?
Yes.
Good. Hi, Nina. How are you?
I'm good. How are you?
Good. We started, so we're recording, just so you
Perfect.
know.
Yes, thank you.
I wanted to see now, we talked about the pathology a little bit. I think you guys are doing all right on that front, right? Yeah, so you're good. So we can go into, I think last week I was, trying to say, let's go do some neurology and then talk about it. Well, we talked about that. Were you able to look at that yet or not yet?
I did take a look at it. It's really dense, I'll be honest. It's
right. That's why I want to make sure maybe we go through the first layers of questions today.
Yeah.
Right. And then we have another sitting on it, because the brain is next, right? So the next week is actually a 2-week module, I think, more or less. We got to go through that, but the brain, that takes me a little long because there's a fair amount of stuff, but it's mostly anatomy. Right? So we have some time to talk about the physiology chapter and how it works. So we can start today with that. Okay.
Okay.
So the best thing I think for that is well, let's see. Do you have any questions to the integumentary and to the endocrine, or is that pretty straightforward? Not right now. Or
I guess like in 3 bullets, how would you sum up? What are like the 3 things you need to take away from those systems? Because they seem like they fine tune a lot of like the balance in our body. And
yeah, definitely. That's
like
a very, very good place to start. Thank you. Let's start right there, because when you look at the bullets you can take away, and this is for everyone. Of course, this is on the video. So when you go there, let's go to test number 3. And oh, well, you know what? This is my bad. Sorry, my background. Let me mute myself real quick. So I'm going to now, sorry about that. I haven't unpublished that for some reason. So I just published this. So we kind of have a whole bunch of information now, and we're going to have a test at the end, right? And it's cover a bunch of stuff. So I just published it. I made a test. I'm sorry about not having done that before. I made a test review like in the previous tests. And in here, you have an anatomy term test review again. I mean, an anatomy terms, some videos, some pictures from the anatomy, like in test 2, but then you also have what we had in test 1, the test review, and you click on that, and that goes to a test review. And in this review, you find, it's like the first test. You have everything listed that I need you to know for the test.
Okay.
So if you, and I'm really sorry I did not, if you, and I got to update this. It says slide. This is old a little bit, but the data is still there. If you go through all those bullets, then you know what we want from that. And for the test, you basically need to just understand on those bullets. You don't need to memorize it because it's open. All right. So we have the skin, we have the immune system there. Look, it's not that much, right? Depending on what endocrine. Of course, the endocrine, we talk about which gland does what, but I wanted sort of bullet points so we'll remember a few things from when we, leave the class and be out in the field, but we kind of remember, oh yeah, melatonin is sleeping and that's the pineal gland or so, it's a few bullet points of that. And then we get to the skin, that the skin is here. So I mainly want you to know dermis, epidermis stuff, a little bit of function, not too much, and what the appendages are, but not too much on that. And then now we started with the nervous system fundamentals, and maybe we take it from there, but I want to make sure we really cover that well in terms of understanding. All right. So how about you guys go and if you go look at those points and make sure those you understand, and if there's questions to those as a beginning, as a starting point for these previous chapters, then you know exactly what I want on the test.
Can I ask you a question about potassium? And wait, scroll down. It's Na and sodium. Excuse me, madam.
All right. We love it.
About sodium and potassium. They're both charged ions. What about what about them both being charged ions make them the same in
yeah, that is one of the things that's a bit confusing. Wait, let me see. I'm sharing the wrong one. I want not neuroanatomy, I want neurophysiology. This one, I think. Yes. No, why is it coming up like that? Gosh. What do you see? Oh, you don't see anything. Do you see anything?
It says fundamentals of nervous system. Electricity runs the nervous system.
Yeah, I get it. I was just sorry. That was my desktop. So when you look at how the nervous system works, I think you bring it head on, right? It's about these charged molecules. It's about sodium and the potassium. And we can basically, we can go and start right here at the nerve impulse and work our way backwards. So I'm going to explain the nerve impulse and you guys help me understand where does something not make sense. Is that all right?
Yeah, that sounds great.
So because you have this sodium and potassium situation right here. So these are both two charged molecules, right? And you carry them in and out across the cell membrane, in and out to the cell on the inside to the outside. So this is the inside of the cell, this is the outside of the cell. Same here, inside, outside. And so when you have a resting membrane potential, when the membrane is not doing anything, it's just still. Let me see, where do we have that? That's here. We have some charged molecules on the outside and we have some charged molecules on the inside. And we have these pumps in the cell membrane that all the time continuously use ATP and pump 3 sodiums NAs to the outside of the cell and 2 potassiums to the inside. They just turn around. There are tons of them around, okay? And then what they do is they push the chemicals around. So they push all the sodiums to the outside and all the potassiums to the inside. And so you have a disproportionate concentration of those chemicals. But those chemicals also carry a charge, and they both carry a positive charge. And so that's where sometimes gets a little bit confusing. And before you have a nerve impulse, what this pump does actually, because it pumps more sodiums out than potassiums or something like that, it creates a resting membrane potential. So it creates a differential between positives and negatives. That's what that means, right? The electricity is this positive and negative stuff. So when you have a little bit more negativity on the inside and a little bit more positivity on the outside, regardless of these molecules being positive or negative. That's just when you compare the electricity, the total amount of positives on one side compared to the other side. That's when you get this voltage, this difference, and that's a voltage. And we call that a resting membrane potential. So that's a starting point. So that's when nothing happens. And so now when you want to have a nerve impulse, is electricity. So you create electricity by sending charges through a cable. In the wall, it's electrons. In the body, we have to figure out that differentiation in charge somehow else. So we use these ions, we use these chemicals. And so now when you want to have a nerve impulse, we're starting right here, we have this resting membrane potential that's a millivoltage. So that's just some charge, some difference. And then what happens is when we get the impulse that, hey, let's fire a nerve impulse, sodium gates open and send all the sodium from the inside, this is actually the inside, I'm so sorry. From the inside of the cell to the outside of the cell. So when we look at here, you have all these sodiums on the outside of the cell. Yeah, now I'm confusing myself. So now you have all these sodiums on the outside of the cell, and when you open the gates to make a nerve impulse, you let all the sodiums travel from a place of higher concentration to a place of lower concentration. It's back to chapter 3. Do you remember that when the molecules cross the cell membrane? You guys remember that?
Yeah. So what is happening? Like, say that membrane is like in your finger or something. What is causing the 2 charges to switch sides of the membrane?
No, that membrane is the one on the nerve.
So if I poke myself on something, is that what sends that charge? Like,
yes.
What?
So if you poke yourself on something, you're going to sever some long extension of a nerve. When you look at did you do the nerve anatomy? Let's go real quick into the nerve anatomy. When you look at the neuron, the nerve cell itself, it's got a few pieces to it. It's got a cell body, and it's got extensions that come out on one side. There are multiple extensions, like tree branches. And then on the other side, it's got one long extension that goes down a long way. And so we have different names. These are called dendrites. This is the cell body, and the long extension is the axon. we're gonna get nerve impulses coming into the nerve from the dendrites and then go through the body, and the axon is where we send a nerve impulse away from the body onto the end of the nerve. For example, that means if you want to move a muscle, the cell body of that nerve is in the brain, and the brain says, hey, we got to move that muscle, and the axon sends a nerve impulse down the spinal cord onto the muscle, and they let some chemicals go on the muscle that then sends a signal and says like, hey, you got to contract, and then the muscle contracts. And so then that's that nerve that sends that signal down. And so the nerve impulse happens along that long extension called the axon. So when we talk about where these chemicals exchange, this is where we talk about along here, but mostly nerve impulse happens in the axon when we describe that. Okay, is that a little more clear?
Yeah, I think it makes sense. It's like they're kind of resting until there's some like sort of irritant
And
that
so then-
stimulate it to switch. And when it does switch, it's along that path at that point. And
Right,
that's the
exactly.
millivoltage.
Yes, that little small difference. And all along that path, this here is a part of that long axon's edge. Like if you think of it as like a hose, long hose, this is the tubing, one side of the tubing. So that's all we're looking at, right? And we're looking at the inside of that and the outside of that, and we compare the sides. Because all we need to have is that difference. And then when we want to send an electrical impulse, we just need to send these charged molecules across that membrane in a different fashion. And that sends that signal. That makes that voltage difference sends the signal that something is happening. Does that kind of make sense?
Yeah, I think what's also really interesting about this is like the fact that here there's ATP again.
Mm-hmm. Yeah.
And it really does power all the different functions of your body. And like, just when you think you know like what the definition of it is, how you make it, there's so many different chemical reactions that use this
Oh, everything.
chemical. Yeah.
And you know what's very interesting though, when the body needs to do something fast and rapid and immediate, it does it in a way that it uses the ATP to establish a situation. Like here, it sends the sodium to the outside and the potassiums to the inside. And then when we have a nerve impulse, all we have to do is open gates, and the sodium is so much more numerous on the outside than the inside. They all want to come to the inside. So then you use a passive process that doesn't use ATP to your benefit, because you can be so much faster doing that. It's like you ride a bike down the hill versus up a hill.
Is that is it reverberation? Was that one of the quiz questions?
No, that's the learning. We got to get to that too. That's a cool thing.
Okay.
That is also nerve impulses, but that's who sends the nerve impulse to whom.
Okay.
That's more the destination question. This is more the fundamental, how does a nerve impulse function question generically. And so then when we look back in this picture, so we take this opening of the sodium channels, right? And so we used this active process to establish that on the outside we have a huge concentration of sodium, on the inside we hardly have anything. And now we're taking a channel that's a passive channel. That's actually it's voltage gated, they call it, and it opens and it just lets all the sodiums in or out. But since there's so many sodiums on the outside, they want to rush in as fast as they can. It's like you have, I don't know, in Tokyo subway or something, and you have everybody squeezed in, and then the door opens on the other side of the next station. Everybody wants to jump out as fast as possible. It's that sort of reaction. And you use that's a passive process, right? And you use that process to fire the nerve impulse. So while you open those sodium gates, all those sodiums from the outside, that high concentration wants to go inside to the inside, and that changes the voltage. Now, all of a sudden, the inside used to be more negative than the outside. Now, all of a sudden, the inside becomes positive because you send all these positive charges to the inside. And that's this spike here.
So is that's depolarization?
That's depolarization. You see the sodium channels are open and all the sodiums go inside. So this is the inside, this is the outside on this slide. And so that changes that voltage. And now all of a sudden we're having this voltage changed, but we need to stop it. But we cannot change chemicals because all the sodium is on the inside now. So the concentration is messed up. So now we open a different channel for potassium. And potassium is also positive, but the concentration is so much more on the inside that's established through this pump than on the outside. And now when we open those gates, everything wants to go to the outside. And so then this happens, all the positive go back to the outside. The positive is attached to that chemical. And so the potassium carries the positivity to the outside and we reestablish that negative inside environment. So the chemicals will be messed up right now, right? The charge is back to being present at being whatever the basic resting membrane potential kind of type is. Does that make sense, fundamental?
Yeah, and the ratio is 3 sodium to 2 potassium as that happens. That
right, and then
cycle of repolarization and depolarization is like a 3-2 ratio.
Yes, and then so you got that change. The inside turns positive because the sodiums rush in. Then the inside turns negative again because the potassiums rush out. And then what happens is the channels of potassium slowly change. Then we hyperpolarize a little. We get a little too deep. We go a little too negative because that's just the nature of the beast. And we call that hyperpolarization. And then slowly, because these sodium potassium channels, they're always working. They're always doing this work. That's what we just, the resting membrane potential. And then they do their work and slowly we go right back to the resting membrane potential level. So 7 and 1 is the same level. The cool thing about hyperpolarization is that gives you a moment while these chemicals remix where you cannot send a second nerve impulse. Right? So you can use that to your benefit at some point later on. Does that make some sense?
It does, and it does. The ratio is never quite balanced, so the process is constantly happening.
Always.
It's just to how far does the the meter swing in one direction or the other?
Right. And, when they talk about the action potential, that nerve impulse is what this mechanism, they call it an electrochemical event. So you have the chemicals that carry the electricity with it, right? So you have two things happening at one point, really. The positives moving around, the negatives, that's the electricity. That's what we really want to send the message. That's that impulse. And we do that by having the chemicals carry that positive charge. And so we need to look at both ends. Like we look at the charge and we look at where are the chemicals. If you get that, you got that chapter. You got that. Because everything else is the rest is just talking about electricity here in the beginning. And then resting membrane potential, it's just what we have. And then when we go forward in that chapter, then it's just about, now it's how are we going to grade an impulse? Let's say, how are we going to make an intensity? Because when we do this process, this action potential, it's always the same process. It's always the same level. There's not like a stronger one or a weaker one. So how am I going to be able to say like, well, this guy really screamed at me versus this was really a soft voice? How can I do that? And we do that by frequency. And that's where that hyperpolarization comes back in. So after the nerve impulse, you have a brief time when you cannot send another nerve impulse, but that's very brief. And then you can send the next one. And so when you need a code, intensity of an impulse, you use frequency to do that. So you rapidly send these impulses as rapid as you can. That would be a very strong impulse. So this is sort of what we're talking about. We have a single twitch, that's actually muscle, but it's the same thing. And a more impulse, because these are the impulses actually, these are the nerve impulses. So you do one nerve impulse in a muscle, you get a little contraction. You do more, you get a little more contraction. Then you do rapid firing, your muscle contracts as hard as possible, it doesn't let go because it really gets that message. Oh, we need to be strong. And so intensity is coded by frequency in the nervous system. And then the last talk here is about the speed of a neuron. How can we determine the speed of a neuron? So we have a few things that do that. One thing is the diameter of the axon, how thick that nerve is. And when you talk about the nerve, when you touch your funny bone, when you hit your funny bone, you're hitting an axon. You're not hitting a cell body. All the cell bodies are sort of as much tucked away into enclosed spaces like the brain or the spinal cord as possible. You got some in the periphery, but you can consider when you hit a funny bone or you touch a nerve, you're going to touch an axon, which is the cable which sends the nerve impulse. Okay, so or an axon, or also actually we have to sometimes also take the dendrites into that, but those are both these extensions that come out, and the dendrites are the extensions that receive a nerve impulse, and they are receiving messages, and the axon are sending messages. So when you have a nerve going from the brain to the muscle, the axon goes from the brain to the muscle. But when you have a nerve bringing information in, you actually have a receptor on the skin, for example. When you talk about that cotton thing, cotton yourself, you have an axon in the skin. That's actually a dendrite, sorry, a nerve ending, and you cut that. In a pain nerve, you really sever that nerve ending, and then the chemicals that happen there send off that reaction that it sends a signal to the brain. It says like, hey, something happened down there. And the brain then interprets what that happens. And so then the message goes inward into the brain. And so the first part of a nerve looks like a full nerve. It is a full nerve, but it is technically going to be a receiving and a dendrite. And then for most receiving nerves, that the cell body is actually right in the spinal cord. That's where people get shingles. That gets infected, the cell body of the nerve, the receiving nerve. That's why they get these skin reactions and these patterns, because these receiving nerves get irritated. These cell bodies get messed up by the chickenpox virus. And so anyway, that's sorry, I went back there a little bit in terms of the nerve impulse of what's what, which direction. But then what we also have to consider is how fast does a nerve fire? So, for example, a pain nerve is a very slow nerve. It fires very slow, because if you cut yourself, and pain is a language of your body that tells you something went wrong and you shouldn't do this, and you don't want that. You want that nerve to trickle down and keep telling your brain that you got to do something about it. And when we say slow, we're talking a few meters a second. So that's like still extremely fast. But when you then think of a nerve that needs to make sure you don't fall on your face when you walk into a puddle and you didn't see it, or something on a hole, and that nerve needs to react really fast, then that's those nerves, those proprioceptive nerves that tell our body where we are in space and then send the signal to the nerve to react. Those nerves need to be very, very fast, and they go about 120 meters a second. So that's very, very fast. And the axon diameter creates the speed of that. And then the other thing that happens is we have a wrapping around nerves, a fat wrapping around nerve, an insulation with little gaps in between. And we call that insulation the myelin sheath. And when you go into through the chapter of the fundamental nervous system chapter, we have both neurons, which are the nerve cells that send the impulse, but then we also have these other cells that support the neurons, and these cells that make this fat wrapping around the nerve fibers, the axons, or also sometimes the dendrites. Those are some of those supporting cells, and those are really kind of having a cool name. They call them the Schwann cells, like the white bird, the swan. A swan, it just called, spells it a little different. But they create this wrapping, and it's of fat. And you think about ions are hydrophobic. They will run away from fat. They like the water environment. That's like the vinegar in a salad dressing. And now you're going to wrap fat around these places where we have these ion exchanges. This is just what we talked about. This is just this. We need to have these ions cross the cell membrane. And now we're inhibiting that. So they can't do that. And so they can't do that around here where we have this insulation. So between each of these insulations, we have a little gap where we don't have this insulation. And so the ion exchange jumps from this area to then this area. And that process, because it sort of has to push and pull, it's kind of like a magnet almost, speeds up the nerve impulse heavily. And so they really use cool names. They call these gaps the roads of Ranvier. And they call the jumping of the impulse, they call it the jumping, they call it the saltatory conduction. So that's pretty cool language, I thought. So that's kind of interesting. Do you guys does that make some sense how that functions? Or why that is?
Yeah, that makes sense.
Is that helpful to explain it again?
Mhm.
Good. So then from there, I don't know if you have any open gaps in knowledge that I didn't touch on right now so far. No?
That seems like a really good start, like a basic what's happening in the nerve.
Yeah, then let me see if I go through this chapter, if we have some blocks in here. No, I don't think so, because this is just the anatomy. We talked mainly, when you really talk anatomy, mainly it's going to be the nerve itself, you know, that the body, the dendrites and the axons. And so that's a good start. And then we can pick it up and do another session on that maybe next week, a little deeper if you want. Would that help? Yeah?
Yeah.
Good. Yeah, no, that way we layer it a little bit. So then let's go into this week's material. Where was that? Safari. Because now this is a big deal, but we're talking about the brain. So I'm glad you brought up the bullet points, you know, because actually I should have opened that chapter before. So now everybody, you guys know the review of all the material we're sort of working on now is in the bottom here under test 3. There's a test review. And so if you can, please go in there and print that out. And then when you go through the material, each time you can review it, you can also go through that list and say like, hey, those are the points I really need to understand, because those are the ones the test will touch on. And then this week we have the brain we talk about. So the brain is quite an extensive organ, of course, obviously. It better be, otherwise we wouldn't be so smart, or whatever we want to call that thing we have. We talk a little bit about the divisions of the nervous system. We have a part that is the brain and the spinal cord, and then the rest is these nerves that go out from the spinal cord to the muscles and to the organs, or to the glands mostly. And so we need to have a sort of an overview of that a little to start the process. Then we get a little bit into brain development. Those are in the movie. So those I don't really touch on too heavily for the test. So don't, you know, just gives you sort of an understanding how basically the body starts developing as sort of kind of almost like a tree leaf or a plant leaf with a stem in the middle and things on the outside, and it's kind of flat, and then it starts folding in on each other. And so you have, when you look at the nervous system, it starts on the 18th day of embryonic development. So it's really, really early, because it has to coordinate the creation of the body, right? So that's sort of its job. And so it starts very early where that leaf, if you can think about it like that, with the stem in the middle, bends in on itself and creates a spinal cord, a neural tube that then becomes the spinal cord. And when you turn that upside down, this is that spinal cord, and at the end of it, you have the brain developing, and that's the development of the brain. And then these parts, they get bigger and they bulge out and they create curves, and that's where then that's becoming the wiggly part of the brain that we see. And that in here becomes, well, and then down here more the thalamus, which is the connections between the two sides of the brain. And then we get into the deeper structures, midbrain and pons, and the cerebellum is the coordination, a lot of the planning part. And then from there it goes down, and this is where it cuts off. It goes down the spinal cord to about level of lumbar 2 level. And from the spinal cord, we have all the nerves coming out on the sides that go to the body and coming back in from out and in, of course, both parts. So that's kind of a little, it's kind of cool how that actually works, or how that gets developed. And then the other next part is we talk briefly about the protection. The brain is very, very wobbly. If you actually take a brain out and gravity collapses, and you can see that, unfortunately, when some people have epidural getting during labor, and it doesn't close off, you take the fluid around the brain and the spinal cord off, and you have a hole that doesn't close, and so it keeps leaking out. And that fluid that is surrounding the brain, so you have fluid surrounding the brain that makes it weightless, that makes it like it's buoyant, like you're swimming in water. And if we don't have that fluid, then the brain collapses under gravity. And often these people, when that doesn't close up for a few weeks until it closes up, they have to lay down much more, because if they stand up and the liquid all goes down, they get a heavy headaches. And so, and it's interesting, one time I explained that in class, and one lady raised her hand and said, like, wow, I had 5 neurologists that I try to get this understanding because I was so horrible after I gave birth. And nobody could explain it to me. It's like, and then I went back to my MD patient and said, like, you know, that was kind of weird. And she said, like, yeah, they don't want to admit these things can happen. It's like, what is wrong with these people? But anyway, don't take that too deep, but it's sometimes interesting what you come across with when you think that would be common sense, and it's not. And so from there, we then go to the cerebral hemispheres, the parts, the cortices. This is the outside, the wiggly part. And when we talk about the wiggly part, the first is a little bit of generic. What happens in there? The wiggly part is the outside. The outside part is wiggly, so we have a lot more outside surface. And on the outside is where we have cell bodies, and where we have cell bodies, that cortical gray area, they call that. Because when you slice it, it looks a little darker than the inside. So this gray area is where we have cell bodies. And the white area is all the action, the connections, those axons and dendrites. And it's actually the white myelin sheaths, those swan cells that do the insulation around the fibers. Those are actually, that's actually the whiteness that you can see. So it's actually fat in here. So brain has, you got to consider that brain is a lot, mostly fat. So be very careful with not giving your kid, fat stuff. Good fat, not crap fat, but, like omega-3s and stuff. Very good, very good. I have an article that talks about the guy who was, a couple of guys. One was poisoned and was nerve dead, and the other one, a car accident, was nerve dead. And they infused those are 2 cases. They infused them at the emergency room, heavy doses, like 15 grams, which is a lot of omega-3s oil into the bloodstream directly. They both come back to life. And so that was very interesting to read that. I have that study somewhere if somebody's interested in those kind of things. But anyway, the brain is very heavily fat. And so you can see that right in here. And so these are fibers. And then the outside is the gray matter. Those are cell bodies. And the cell bodies is what we consider where we make decisions, where the brain thinks, and the whiteness is the wires where the impulses get sent to and around. And if you do a math problem or you study some anatomy, there's a lot of connections that got to connect to each other. That all happens, that we can remember that. And that, Nina, goes back to that reverberating cycle real quick, because if you do like a problem like memorizing something, you have a neuron after a neuron, and they all talk to each other. And one of those connections is what creates the solution of finding the right answer, for example. And the more you strengthen that, the more you repeat that connection, then nerves at the end of the connection fire back to the beginning of the connections, and it strengthens itself. And that's called a reverberating circuit. And that's how the brain learns. And so that single understanding lets you understand neuroplasticity and everything. So at some point, I want to make sure that you understand that because you got all of you, because that's how the brain works. That's why when we continuously think about negativity, we're gonna be depressed. That's the gratitude journal that we'll do this next week's is about that. So anyway, that's in the thing, but I just want to mention that because you brought up that reverberating circuit. And then from there, we go into the lobes, and we have mainly the different parts of the brain outside part, not the inside of the brain, the outside part. And those are structured in lobes. And we have a frontal, a parietal, a temporal, a occipital. Look at that. The same names as the skull plates. That's perfect. Here, except we have one deep inside. If you separate these out, let me see if I have a picture of you. Nah, I can't have it. I don't, in the booklet, there is a picture, but on the deep inside, there is an insula. And so it's deep inside here. When you go and pull those apart, inside, there is an extra lobe. So it kind of folds in on itself. And you can see that sort of here. You see how that folds in on itself, that in here, this is called the insula. So it's on the videos for sure, but you got it, that every lobe has sort of a main things it does, and we'll talk a little bit about that. Studying the brain is quite difficult. It's not like the heart where you kind of make a full story and then it all works. It's more like in the brain, it's like, oh, the occipital lobe has to do with vision, the temporal with hearing, and it's a little bit more chopped up. And unfortunately, it's hard to get, it's hard to study the brain because we're still really learning a lot. We don't know that much. And so we're giving you sort of a sketch of what we know, or more or less of what we know. And so learn it as such. And then when it comes to certain aspects, when we get to the spinal cord, I talk more about how the one talks to the other and so forth. So the lobes are those. And then when we go deeper, this is the lobes, right? So this is the insula again. Then we have structures deep inside, and I talk about those a little bit. Diencephalon is deeper inside, and then brainstem and cerebellum are deeper structures down here, and it just basically talks about what they do. So just listen to them, print out the test review, and kick back and let the learning process take place without too much of the anxiety. You don't have to memorize these structures, you just have to know how they kind of work and how, what works together. Okay, and then here, after the brain discussions, in this and the videos, we have questions, we have labeling, we go back to the blood pressure, which you're probably finishing now or have finished. Do a little talk. How did that work for you? Was that hard or easy? Just a big back and forth. And then we have a thing this week where I want us to focus on positivity. So I have a TED talk to listen to, and this has back to do with the reverberating circuit. Because if you want to be happy, you got to be grateful. And so this talks about that. This priest, this Austrian priest, has a really funny accent. And then I want one exercise that UC Berkeley has established. There is actually a greater good in action department or something that looks at things to make our lives better. It's really cool. They figured out the research that, like, for example, if you stand in line and you talk to people and do small talky stuff, that's actually really happy making for people. It's very important for the contentment. And so they have stuff like that. But one exercise they have is this gratitude exercise. And what you do is you, at the end of each day, you think back of your day and you find 5 things that made you happy that day, that made you smile. And it can be as banal, as simple as possible. It can be like, oh, look at that. I looked at the sun and it was just a pretty sunrise or a pretty sunset. Or, wow, we got some rain. I was so happy for a moment. I know maybe we don't get smoke this year, or whatever it is, right? Stuff like that. And then you write them down. So you do at least 5 things and take a moment of reflection and just remember back and then write them down and then cherishing those moments, a brief moment, because we have found in Buddhism and studying and psychology and all that, that if we do that, it's like those channels open more and we get happier over time. And so the gratitude journal is basically what that's called. I want us to do that for 1 week or 5 days, I think. We'll take 2 days off. And that's not that long, but I've seen with a lot of students that we're already having some kind of response on the inside if you're doing that seriously. So if we do that as a unit, that would be great. And then after that, we have a brief discussion around it, like with all of those kind of exercises. And I think that due date again is 1 week out, because I don't assume everybody starts with that today. I assume by the end of the week. And that's it. And then, of course, a little Padlet at the end. Yeah, some people I see use the Padlet to communicate with their groups. Oh, yeah, pathology groups. You guys are doing all right with that? Yeah?
Mm-hmm.
You guys moving? Good. Okay. Yeah, I talked to you a little bit, but make sure everyone reach out to me and I'll be in touch with you guys over text, and I'll try to, keep coordinating where there is challenges with conversations, but I think so far so good. You guys, a lot of you are moving right along, so I'm happy about that. Okay, good. So that's this week. Is there any questions left over, or is that pretty good?
Yeah, I have more question regarding the brain.
Mhm.
Yeah, so when you were talking about now at the end of the day, you do journaling and you start to write down 5 things that make you happy. So so am I right that the part of the brain that decide to do that is the frontal lobe?
Yes.
Okay, so when you start making that decision, that started from the frontal lobe, and then
Yes.
you recall these memories. When then it make the connection, the neurons gonna make connection to what part of the brain that hold these memories.
Well, ultimately, the part of the brain that's a very good question. Ultimately, the part of the brain that holds the memory is going to be the hippocampus. Let me see. Let me see if we find that part here. I didn't pull the brain PowerPoints up, otherwise I need to do that. Pons, brainstem, midbrain. No, hold on. You got me there out of the thing. I got to find it. Frontal lobe. So frontal lobe is where you make the decisions and all that. And then you got all these other lobes. But where the heck did I put then the memories? That'll be the limbic system. Basal ganglia, limbic system here. So the limbic system is the emotional brain, which is very interesting because that's the brain where you're happy or sad or feel, you know, there's the anger center, the amygdala, like that talk about the amygdala hijack, which is where we find, you know, amygdala is where we feel fear. That's deep inside here. Where was that? Amygdala here, this piece here. So there's a structure inside here. The amygdala is where we feel fear and then react. So that's like if somebody has a freak out and a temper tantrum, that's in there. And so they call that in neurology, they call that an amygdala hijack. I have that somewhere in the book too. And so that's where we have the emotional, and then the hippocampus is right here. And that's where we make the memory. So that's where the memory physically will be stored in here. All right.
Okay. So when you say it's physically stored in there, that are they like just neurons, the same things at frontal lobes material?
Yeah,
are they
neurons talking to each other.
Okay.
Neurons connecting to exactly how and all of that. That is actually a very good question. But the only thing with that I know is we have connections. They are connected one to another. And the connections are what is actually most crucial in the brain stuff. So we learn what's where, but the connection with one another is most important. So some people say, like, if you get dementia, you don't find the memory anymore. The memory is still there. You just don't know how to find it.
Okay. Yeah, that's true.
Right. And so that's that hippocampus. And the other thing that's interesting about that is you can't study when you're depressed. You can't memorize. Your memory center goes down. When depressed people have less ability to do that. And that's a physical reaction inside the brain. So it behooves us. It's very important to try to create as well of an environment as nurturing as we can to do these processes.
Yeah. And I noticed one thing, I say, if we have a emotional connection to something, it's easier to remember it. And is that so? Physically, the hippothalamus is next to the amygdala. So physically, they are connected to each other. That's why. Is that why we tend to memorize something better when we have a more emotional connection to it?
Absolutely. No, that is a very good point. Okay. That is how it works. And also, you know, the rush, you have to kind of make that environment. I know for myself, the cramming down, I have to study the way that I can study. My brain can do it. It's comfortable. It's not, I can't cram for a test. It doesn't do anything. It behooves us to do that. So yeah, that's why the exercise this week is I want you to do a little bit of that. Most of you probably already do it, but a little bit of that gratitude, that exercise of remembering in the evening, and see if that, you know, those few minutes See if that makes a change. Cool. Anything else? Any other questions that come up right now? And if not, you can always text me if they come up. We can always talk offline too. There's no problem with that. Okay. Good. With that, if nothing, I send you your way for the week. Okay. Any last thoughts? Are we good?
We good. Thank you.
All right. Have a wonderful weekend.
You too, Dr. M.
Oh, no, week. Take care. Bye-bye.
Bye-bye.